Hello everyone. Thanks for coming this session. This is about spatial technologies, but with an emphasis on experimental technologies. So I'm Xiao Wang. I'm an assistant professor at Broad Institute and MIT, and our lab is working on developing all kinds of imaging-based spatial omics tools. And it's a great honor to be here to lead this discussion. And yeah, and with that, I will switch to our co-chair here.
Hi, I'm Kazumasa. I'm doing a postdoc in Sarah Teichmann's group at Sanger Institute. And our group is mainly working on atlasing of multiple organs using multiple modalities of the technologies, including single cell technologies and also spatial technologies. And also we do some computational pipeline development. But yeah, it's kind of many things we do for the atlasing project. And for me, I'm working on HART project using spatial data as well, mainly Visium. And I have a poster there, number 12. So if you are interested, please come. And our paper with James Cranley there will be published in Nature this week. So please have a look if you have a chance. So yeah.
Yeah, great. And then as far as we know, like this room is equipped with like all these microphones and the cameras. So if you have any questions or if you have any comments, you can just stand up and speak. So you don't need any extra equipment. It's just like with your hands and always stand up. And in addition, I invited Fei Chen here. You have listened to his talk. So there's a lot of fancy like sequencing or array-based spatial omics tools from his lab. So if you have any questions, you can also address to him. Okay.
Okay, I will start a few slides about this session. The first slide is about the summary of this session. Mainly, so it's also in the web portal, but main thing to discuss would be there are various spatial technologies and there are trade-offs for each technology. So it would be nice to highlight those trade-offs and pros and cons for each technologies. And so basically, I will start with the spatial transcriptomics because I think that is the most popular technologies in the field at the moment, which is basically split by the array-based and image-based technologies. Array-based has this Visium, DigitSec, SlideSec, StereoSec, those technologies, and also there's a slide tag which Dr. Chen Fei presented this morning. And the resolution is really various. It's from 0.2 to micrometer to 50 micrometers. So there's a lot of difference in terms of the resolution that you can capture. On the other hand, image-based is can also categorize into in situ sequencing or multiplex fish. And also the difference there is you can either be targeted or untargeted way. And the beauty here is you can have a molecular-based resolution. So it's much higher resolution than the array-based technologies. And other technology assign Zoom. Yeah, maybe. We're fine. Okay. Okay. So either technology needs to assign measure data to single cell level, to be single cell type level, to be able to do the downstream analysis. And for the array-based assays, because of the size of the spots, I mean, this is array-based technology, so it cannot be like single cell-based data. So this requires the cell type deconvolution, and there are many pipeline at the moment, and you can choose. And that will give you the estimated cell type abundance or proportion in each spot. On the other hand, image-based assay requires cell segmentation, which often requires manual fine-tuning for each tissue type. But also there are some segmentation-free methods like Baser, which was also in the morning session, and to be able to assign each molecule expression to each single cell type level. And after that, you can do various analysis, including collocation, spatial neighborhoods, and cell interactions, fractal annotations, and spatial bioregimes, and cell type spatial region process, and so on. I will switch over to John.
Yeah. So and then spatial transcriptomics is very cool, and but there could be other more information being gathered through spatial technologies. I think there's already like multiple methods developed, demonstrated to profile the genome, the epigenome, histone modifications, DNA modifications, or chromatin accessibility. There are also new methods towards the downstream after the transcriptomics, such as the translatome, proteome, metabolome, and the cellular functions. So I think we will have a few questions about how we can actually better integrate those multi-omics information to some extent, because we have extra spatial information from all these spatial technology platforms. It may be even easier for spatial technology to integrate than like a single cell technologies. So, but we want to point out the foundational biological questions and the topics that all those spatial methods could provide, such as at DNA level, we could trace the lineage. We could do somatic mutation, a lot of DNA sequence analysis, and then at the epigenome or chromatin accessibility level, we could better understand the epigenetics and gene regulation network at a single cell resolution and within spatial context. And with the translatome and the proteome, there's another level of gene regulation happening at the protein level, right? So control the synthesis and degradation and the localization and the modification of the proteome. So there are also a lot of very interesting and exciting biological questions behind the translatome and proteome. And finally, we want to gather all this information to like in a convergent way to better understand the gene to function relationship. So also with the recent development of other advanced mass spec or imaging-based approach, we could potentially spatially profile the cellular metabolism, mechanics, physiology, electrophysiology, and also many like functional medical level of imaging. So I think that's another very exciting aspects of spatial technology. Is not just for omics, it's also for functional imaging. And in the top for like biomedical topics, it will be so fascinating. We could integrate omics with imaging. That's the new opportunities we also hope to cover today. Yes. So with that, we just want to switch to the questions. We try to organize all the questions into 8. I think we start with 20, 30-ish. But still like, let's try. We hope we can cover all of them, but basically there's like 2 types of questions. One is how we can utilize those spatial technology to build the atlas. And then later on, it's how we can use them to better understand such topics such as gene to function, gene regulation, and as well as like human disease. And we try to follow the order, but if there's any like relevant discussion arise, we can change order as well. So with that, probably we can go. We can start with the very first question, which is the choices of a spatial platform, which may be harder choices than single cell omics, because with the spatial, there's always like there are different experimental setups. There are also different emphasis on resolution or throughput. There are also choices of a selected gene panel or whole transcriptome, and also probably you also care about the general sensitivity and whether they are compatible with other imaging modalities. So probably with that, we start with the choices of a spatial platform. And if you have any questions or comments, please just raise your hands or stand up.
I think that in the end,
sorry, would you mind to have a very brief self-introduction if you are asking your very first question?
Okay,
Yeah.
So my name is Nadav, and I gave a talk this morning again in the spatial session. Actually, my background is neuroscience, and like a lot of I've done like it's like 10 years of a lot of imaging and kind of like image processing platforms from there. And then I moved to immunology to actually kind of implement a lot of these different things. And I'm actually like my like in the end, like I think that we can all agree that the basic unit that we want to investigate is the cell as and in that sense, the resolution, like the question of resolution is kind of like I don't like, I think that we focus on it a bit too much, and the question should be, can you capture the cell and can you not capture the cell profile? In that sense, for example, for neurons, even if you do capture the nuclei, do you actually capture the cell? Because all the interesting processes are actually happening in distally expressed genes, and 50% of the genes in the neurons are actually differentially kind of like locally transcribed to the synaptic region. So in that sense, kind of like we know already, like in the mouse, like layer 1, layer 2, layer 3, and all the cell types, it's great, but I don't feel that it really helps us understand the brain function, like in that sense. And in immunology, can we actually capture, like I translate the question to actually the only missing kind of spatial modality that I think is really challenging in spatial to get is like the surface receptor like array that we get from sightseeing, for example, which we have found that is extremely useful specifically for the thymus, like to understand what the actual cell is doing, because everything is done on the level of whether a receptor is internalized or expressed on the surface. And often in imaging, whatever modality, you kind of never get that information. It's even difficult for me to imagine how one would be able to get that information, like using this. So just, like, how do you see that?
Yeah.
You think adding the protein level is actually really important to be able to?
Yeah, I think that what I'm trying to connect is like kind of like what defines a cell. And I would say that a neuron is defined by its synaptic connections, and our immune cells, it's also largely defined by its cellular interactions.
Yeah.
And in both of these cases, we don't really have that information in the current like spatial modalities.
Yeah, I think that's a good point, right? So for neurons and immune cells, they are probably two most communicative cells, right? So they talk with each other through synapse and other cell surface receptors and interactions. So regarding the question 1 about neurons, right? So I think we have developed some tools to allow people to spatially profile mRNA translation, hopefully to restore that subcellular information to better understand, for example, synaptically localized mRNA translation. But that's not enough, as you mentioned, right? So we have to actually overlay the connectome information.
Yeah.
So I think there may not be an easy solution for neuroscience, but I believe there are many labs trying to develop, tracers, right? Transsynaptic tracers. Although it's still a very tedious way, but I think if we combine, for example, imaging-based neuronal tracing, especially transsynaptic tracing, and overlay that with either spatial imaging-based spatial array or hopefully sequencing-based spatial array, I think that will be extremely helpful. But I feel that's kind of need a lot of domain knowledge and targeted development to targeting the connectome. Probably an easier one is some glia neural interactions or glia cell interactions, which is already happening in many labs, right, to study Alzheimer's disease and aging, to see what has been changed in these glia neural interactions or glia cells. And for immunology, I think they are like a spatial sightseeing and other type of work, try to co-profile protein versus RNA. And regarding your question, how to differentiate the cell surface versus inside the cells, I think that's again a harder problem. I believe probably people could do some perfusion to use some reagents that don't enter the cell, so they have to stay on the surface of the cell. I think again, this may need some really specialized development to differentiate intracellular versus extracellular space. And yeah. Any other comments on this topic?
Do you mean when you say the surface receptors? Do you mean what do you think of sightseeing data for that?
So we've seen, for example, that specific that there are specific kind of surface expressors that you can see, for example, like even TCR receptor signal or CD3 abundance, that you can really see that you have different subtypes or different proportions, kind of like a bimodal distribution there, but on the transcriptomic level, you have nothing, absolutely nothing. Like there's no correlation, you don't see anything in the human. Like in terms of when you look at the RNA profile in single cell.
Yeah.
Whether you would see it if you measure the protein, I don't know. Like the protein from internally, I don't know. But
Well, sightseeing Generally, it's surface.
Yeah, no, I mean like if you, because in spatial, there are spatial techniques obviously that you measure protein in general, but there you can differentiate between the internal and external portions of like whether a receptor is internalized or not internalized in that sense. Or for example, when you look at the Nexin-5, like it's clear that like just measuring it doesn't really indicate like whether the cell is actually dying, but whether the cell is expressing it on the surface. It's like a better indication. Of that.
Yeah, so I guess probably with that, I think we can also briefly mention about this atlas integration thing, right? So I think although probably it's still hard to use one experiment to measure everything all at once, but it's potentially possible to take the same sample and cut them into different chunks. And one slice could be the spatial transcriptomics plus protein imaging. The other chunk could be site-seq or other type of single cell and other modalities and computationally restore everything, hopefully at once. So yeah, I think maybe eventually given more spatial technology happen and across so many different labs, maybe tomorrow's session about computational integration will be helpful.
I guess one key point is how can we leverage the single cell data to spatial data? If we, in ideal case, if we could infer many things from the single cell data, which is single cell based and may have more high quality, like high depth data, then that will change. That will like upgrade the spatial data. On top of that.
There are questions.
Yes.
I think the point you raised last, this is very much chapter from Indiana University. That's what we did with our spatial transcriptomics. We initially did the single cell RNA-seq data, and from that we derived a couple of signatures of the epithelial cells. Then we took out the and modules of the breast and applied those signatures to figure out what is the
another way to go him down. We went down on him. It's the way to go him down.
Can people on Zoom
Found
mute?
that online.
Can people on Zoom mute, please?
He slept in the shade all day here in the guest room. I think that's his favorite spot now.
I should.
Maybe put it in the chat too.
Yeah, maybe you can mute them.
Yeah.
You can mute them because
Yeah,
they're
you
hosts.
can.
Yeah, this room, right? So there's another room, like we just mute this room.
This is our room, right?
This is our room. Okay.
How can I mute?
Okay. Yeah. So how to do this? Like, we can keep the discussion while figuring
I
this out.
can help figure this out. You
Like
guys
this
can-
is our room, right?
Yeah, you can just
Sorry,
figure out
could you-
who it's.
Yeah. So
Oh.
that's what we did to figure out what are the genes uniquely expressed in the globular epithelial cells versus the other epithelial cells in the breast by imposing single cell data over the spatial transcriptome.
Yeah, but what I'm talking about is a situation where you don't have that information. So obviously we can uncover a lot of diversity by doing that. And in certain modalities, so we see it in our data in site-seq, we also see it with VDJ. So where you can really nicely like model the VDJ data that we don't have yet, but we don't have a spatial VDJ. And that's completely orthogonal. There is no way you can infer like a VDJ from the RNA. Like no way. And so I mean, you need to measure it, but there is like if you don't do like a specific measurement for that, then yeah. So I guess like my question is like more about like in cases where you cannot discriminate. Like there's the
Can I ask one question to Dr.
Yeah, sure. Sorry, we're not hosts, so we can't actually mute them, and we can't claim the host. So
Yeah, we just need to speak louder.
you can only ask them to mute themselves.
Yeah, but they're clearly not listening.
It means
Yeah, sorry, go ahead.
By hearing your session in the morning, I felt like the strategy is really like covering what we discussed now.
Yeah.
You tag based on the, and then you can go to the single cell based analysis with any modalities. So,
Yeah. Yeah. Sorry, but in the question.
so the question is, do you think that technology will overcome any technology exists? And
I don't think so. I think, I think though there is something that I alluded to at the end, which is that many of us who are collecting atlases are collecting paired single cell data. Many times single nucleus data, just because of and it's really the only reason we're collecting single nucleus data in many cases is just we can't collect single cell data from frozen tissue. It's not because intrinsically, we want to collect single nucleus data. It's just, it's like a sample limitation. So there's a lot of collections of single nucleus data. And we should, if you're already collecting the data, you might as well get the spatial information associated with it. Right? It's not any additional work and no loss in data quality. So that's one thing that I think is important. And the other thing is that we talk a lot about computational integration, like there's cell location, RCTD, these like methods for doing computational integration on the spatial transcriptomic data. Those methods are pretty good, but I think you leave a lot on the table doing deconvolution, especially when you're thinking about like cell states, and you don't leave that on the table with intrinsic single nucleus data, or single cell data. So I think that's the other component of it. I think it's going to be really important.
Because you really want to have the spatial data as one of your variables to do the UMAP, to do the subclustering, right? So that that can feed straight into deciding on the subpopulations or substates.
It's just that the substates often, it's hard to do the deconvolution because if you think about what the substates are, they might be like that, like two cell subtypes could have some vector that's shared, right? Like activation, right? And their deconvolution algorithm will not be able to deconvolve that no matter what information you give it, or it's going to be very hard. And so that's one challenge, but also just integrating spatial information into the clustering that's a totally different and much just as interesting problem. But I'm just talking about the existing like strategy that many of us here are using, which is like, let's collect our single cell atlas and then let's collect our, slide sequencium, whatever data sets, and then integrate those together. We do still leave quite a lot of stuff on the table for the integration.
Yeah, and also like there are like a lot of the subset, because I'm more RNA biologist, so like there are like a lot of subcellular interesting thing happening like in the synapse at the ER and at many like cell junctions. There are a lot of interesting RNA and protein biology happening inside the single cells that we will just lose that information. So I think in the end, if we could have some like multi-omics integration so that we first have genomics and epigenomics and nucleate information to have a global picture of what's going on with the cell type and some permanent epigenetic change, but we can also overlay a high resolution like subcellular events that will be eventually also helpful. So yes.
Just quickly, I don't know if the people on Zoom can hear us. Because I know when I was on Zoom, I could only hear it if people spoke into a microphone.
So I asked that question. The answer is our room is full of microphones, so they should be able to hear you over Zoom.
Okay.
That's what I was told, so I haven't tested.
But can someone on Zoom say something if they can hear us?
Yeah.
Well, they muted themselves, right?
Right. So
please let us know if you can hear anything.
you can type in your questions, or you can type a yes or something. Yeah, I don't know. So, but it's recorded, right? So,
Thanks. While waiting for that.
We can hear you. Yeah. One of the audience
oh,
said.
great. Yeah.
Okay, fantastic. So I just wanted to comment on the integration question. This from sad personal experience. We had a paper in review where a reviewer told us, our methods were outdated, and don't we know the new way of doing things is to do all your cell state analysis and single cell data and simply do label transfer or deconvolution. Like, don't analyze the spatial data for clustering or anything like that. And I just want to say from our experience that doesn't work so well, because the single cell data are clustered in one manifold based on one set of feature genes. The spatial data may not have the same genes detected, they may not have the same feature genes. Then essentially you're looking at the data in a different space. And you're going to see different things. If you try to shoehorn your single cell states into that, you're just guessing very often, because your spatial data do not have the gene detection to really separate those single cell states, and then you're just flipping a coin and you get something you think you found that sub type in your spatial data, but you haven't, right? So I just want to make a pitch for, it's not enough to just, single cell data won't give you all the states. You have to learn it from the spatial data also, or some kind of balance. So I'm curious to know how people balance those two and end up with this cell in this spatial data set with this up.
So I think one answer maybe you just try to measure everything with spatial. In the end, either use select type or other imaging-based transcriptomics. So I think in the end, it would be nice to actually probably using the spatial atlas as your reference. And if you want to restore some information that cannot be measured spatially, you measure single cell and put it back, so that I think, yeah, if we could experimentally directly measure everything in situ, that's the best, right?
How far do you think we are from that?
I think it's not only a mission of academic labs, it's also like how far it's going faster and cheaper. So I think probably next year we will be more and more such thing.
The problem is robustness, I think, like in many cases, like Visium is fairly robust. Like in our, and this is why it's so, I think it's so popular.
I think it's
okay,
also
so cheap.
Sorry,
by
I think
spatial
I'm
and
muted.
in-situ approach, yeah, could be
Let
actually
me
cheaper
check. Where's
and easier to
my?
handle.
Sorry.
I mean,
Yeah.
it's like we've had like, so it really.
So I should bring.
The thymus, we couldn't get certain spatial technologies like to work at all. But for fetal, it does work. So it's really like specific for.
Tissue properties.
Yeah, some tissue properties that need to be adjusted, and like these companies, obviously, like even 10x, they don't test all the different aspects. And we actually realized at one point that
It needs us. You should mute yourself.
permits to get like better representation of the data. But like, for example, like, but when we tried, for example, Curio, it worked actually quite well. So it's like in different. I think that again,
Optimization.
yeah, but it's much more expensive to do Curio. Like I think it's like 5 times more expensive in terms of sequencing.
Oh,
So,
I see.
yeah, maybe we can move to the next like comment.
I have a question about the nuclei tagging and linking that to image segmentation. For example, the size of the collagen fibers in fibrosis or the size of the lesions. Because in healthy liver, you can basically sample any liver and they just look beautiful. And what we're seeing in disease is the size of the collagen deposition and then the size of the lesion and even the edge of the lesion, like whether it's diffuse or sharp, that's changing the microenvironment.
Yeah, no, that's fascinating. Yeah, I think that's something that needs to be, it's doable, but it needs to be developed and just needs bandwidth, either on a commercial perspective or an academic perspective.
And do you think it will require serial sectioning or would it be done?
I mean, ideally, you'd want to do it in the same section. And serial sectioning for something like collagen could be maybe good, but yeah. I actually wanted to return this, is that okay? Can I return to this comment about the integration really fast? Because I think it's important in the sense that I think there's a lot of, because of the, for some of the methods like Visium, they're like robust, there's a lot of data out there and there's a lot of single cell data out there. So I do think the integration is important. But I think there's not a good, maybe there's just an afternoon session on computational spatial methods. There's not good, I wouldn't say benchmarks, but there's good benchmarking data, but people, we don't have like, here's the level of resolution that any given integration method support. We're very ad hoc. We developed an integration method, Omer developed an integration method. We compare sometimes on the same data set, but if you think about it, the integrate, like when you go to subtypes and these other sorts of things, there's no like standardized test metric and across data sets. So that could be really valuable for just evaluating.
Yeah, some have some ground truths.
Have some ground truths, yeah.
Okay.
Okay, yeah, I'm a PhD student in Jesse Gomez's lab in computational biology here in Toronto. Okay, so I have some thoughts on integration, but reading papers is like so much stuff on integration.
Yeah.
And to kind of give you my 2 cents on it. First thing, I think it's a very contingent problem. And by that, what I mean is it's contingent based on technology. If we could have the whole transcriptome and single cell resolution, nobody would care about single cell RNA sequencing anymore, because then you would get the same data plus a spatial dimension. That's like, better. That's one thing. And the second thing is, I think, there's a lot of multicollinearity in genes. So now there's like a whole, the problem where people talk about, selecting the minimum number of genes such that they capture the variance across the whole transcriptome.
Mm-hmm.
I think, before we're talking about, which gene patterns you select and whatnot, to some extent, that's a question dependent. Is your goal to discover new cell types? If that's the case, then select the gene panel that captures the most variance of the whole transcriptome, because, that's like a way of sort of capturing the subtleties between different cell types. If your goal is to these are like important functional genes or whatever, and then you want to see 2 different cohorts, how they differ, and do something like differentially expressed gene. So I think the question sort of drives that. But I really think in like 5 years or 10 years, nobody's going to be talking about this. So for example, earlier we saw the resolution is getting smaller and smaller, and if somehow it can get to not subcellular and not like extracellular, but sort of just at the cellular level, then I think integration sort of like fades away. So even though people can write papers and get citations or whatever, I think it's not like a universal problem, I think. So I would sort of put my money on the technology development side of things, that that's going to solve the problem, not the computational side.
One thing, I think the difference of suspension data and spatial data is that the size of the tissue you can capture. So I think like if you go with suspension data, you can have a nice representation of one organ or something if you sample very nicely. But spatial data, especially for human organs, it's really hard to capture the whole view. So I think that is yeah.
Yeah, that's a great point. Is like whether you got a shallow sampling of a larger volume or you have a concentrated sampling of one of those organs. So yeah, so I think that it's great discussion about integration. But the one thing I just want to say is like it's also about the spatial to spatial integration. Just as I mentioned, like it would be nice to have spatial multiomics through this, but we can go back to the second point of spatial multiomics. But I want to move forward with this 3D mapping, reconstruction, and also this common coordinated framework, because that's kind of like maybe the ultimate goal that we do have a like spatial atlas of the human body. And there besides what we can measure, I think this, when we talking about the 3D reconstruction and the CCF, probably we also need to talk about what's the volume we are talking about, right? So how we can evenly sampling the human organs as fast as we can, but with a good coverage. And yeah, so probably we can switch to that topic to make sure we can finish the whole list.
Maybe we can have a comment from the lab.
Yeah, I can start with a controversial statement, but I don't think you need 3D data. And this is coming from a person. I've published manuscripts of 3D data and done a lot of light sheet microscopy. And it's not completely true. Obviously, it's going to be like for specific vasculatures and these things, if you want to, then you actually need like for the micro feature. But for the global feature, I think that if we know our organs good enough, then we can actually draw these CCFs to be very accurate based on the 2D information that we have. And we actually don't need to align physically, like the physical space, we don't really need to align. We need to align the macromorphological features of the tissue. And then we can also get like a higher dimensional representation. Like it's not like that the coordinate framework needs to be limited to 3D. It needs to be, it's ND.
Yeah.
It's actually ND. What the cell is seeing is an n-dimensional space of cytokines, of other cells that are interacting with it. And actually, I don't think you need 3D. Like, and I think that the data sets that we have will never be 3D. Like, because it's just very, very complicated and it will cost a lot of money to do. And honestly, like, from all the attempts that I've seen, all the papers that I've seen that actually measure 3D, I haven't seen even one example where they could draw a different conclusion based on having true 3D versus like versus 2D. Like, I mean, and again, of course there are exceptions. So I'm just like, know that it's like, so just to stir the discussion.
Yeah, I have different opinions from you, but I totally agree with your points. Like, I think probably a large fraction of biological questions or what we care about can be totally addressed in 2D. But there could be some utility of 3D. For example, if we have this medical imaging of MRI, and there are a lot of like large volume 3D whole body or whole organ type of imaging, we actually want to register our spatial like 2D data to some, correlated.
But why? I mean, why? Why would you want to do that?
Because there's a real need for that. Like,
But
I don't know. I'm just speaking from like probably more neuroscientist background. I'm not neuroscientist.
But again, in neuroscience, so the neuroscience is a perfect example, like the atlas, like the.
That's where 3D is necessary, and also because people do in vivo imaging, like calcium imaging, and a lot of like directly in vivo mapping, they are all 3D data. We have to register the spatial omics with that intact 3D data. That is very hard to do with.
But in this registration, especially in humans, you're losing a lot of resolution. So you're trying to overfit a biological sample to a reference atlas exactly in the exactly same coordinate. And for the mouse is quite, consistent. Humans are very variable, as you know, like in terms of the kind of like for neuroanatomists, for them to know like which region, they need to do a lot of, like warping and alignment for human, and they also need to do another layer of functional imaging just to infer the region. Like the anatomy is not even enough in many of the cases. So sorry.
I think it actually really depends on the morphology of what you're looking at, right? So we work a lot in the gut, and in the gut you have a tube, right? It's a really long tube. And really we register because what we'd like to find is landmarks that aren't physical landmarks, but actually molecular landmarks or cellular landmarks of where you are along that tube, right? Large intestine is huge, right? It would be really nice to know where you are along that axis. So actually registration there for us is critical because we really want to understand where the gradients are along the.
Couldn't you infer where you are along the tube from a composition of morphological landmarks that you have in the section?
No.
Are you sure?
Pretty sure.
No, I mean, like the thing is that these things were not tried. Like people didn't attempt to do.
But I mean, we have imaging along the way, right?
Yes, but did you try to quantify like the local distances between micromorphological features in that gut tube to see whether I'm sure there is. If there is a difference, then there is also a difference in the thinning, in the thickness of the sections, in the kind of the micromorphology even of the cells that you see there. I would expect that there would be a difference that you could predict from that. And then I
guess you have to measure it once, so you know. Then in the future you may not need, but you have to do it once.
No, you have to.
Yeah.
You have to know your organs. You have to have you need to have the 3D to build the reference.
Yes.
But then like what I say that you from that point when you know what are the features that you need to look at, what are the landmarks, then you actually don't need to register it physically anymore. I
kind of agree with that. So I feel like my point is like we need to do it at least once, so we know the correspondence, and in the future we can decide, do we want a like molecular registration or physical registration or some sort of combination.
So should we maybe start out with building really detailed H&E atlases? Because actually it's cheap and you can really get insight, and then you can also do reconstruction of the 3D structure from just that very simple morphology.
So actually in the gut we do 1D, 2D, and 3D, right? First it's distance along the tube,
Yeah.
right? Then it's 2D where you are in terms of, along the gut, and then it's in a 3D reconstruction model.
But
So,
3D
and-
reconstruction is many 2Ds. You don't collect the 3D like light sheet, or do you collect
Well,
it, like,
MRI gives
MRI,
you the 3D data, right? And then along the MRI, you can actually construct the axis, and then you can correlate that with the 1D, distance.
Yeah.
Because,
But then
honestly,
you keep track of where you sample in 2D, right, for that.
Yeah, I think it's like typically it's like we try to have a imaging based large volume thing to at least know the landscape. And then there could be sparse sampling in depth molecularly, omic scale at a selected position, but we can register them. And yeah. So yeah, but I can agree with your point. It's not a 3D problem, it's an ND problem, right? So it's like you have 20K gene plus 3D, so it's like multi-dimensional information. So probably in the end, like molecular or gene expression-based search engine or imaging registration will be even easier than a traditional imaging-based alignment.
Yeah, so one point you raised was like, what is the reason or the motivation behind 3D? So, I think I would draw the analogy with like functional MRI. The reason people do it there is so that, the brains of two different people are kind of different on the edges.
They're really different. They're extremely different.
So you want to say, my temporal cortex and your temporal cortex have these differences, but are those differences these contingent differences in shape, or are those differences driven by the phenotype you care about? Because of that, you would want like a 3D, 2D atlas. So, in the Allen Institute, what they do, I think the 2D, 3D question is like a matter of sampling depth. So they took I remember this one, 1675 mice, 2D sectioning and using that to make a 3D atlas. And then when you do, spatial transcriptomics, you register your own samples to that. So then you can say, given the average representation of the mouse population, the sampling distribution, the temporal cortex of our species, it looks like that. Okay, and then you can compare different samples between different groups. And that makes sense because otherwise, the kind of signal, the difference you observe between the groups might be driven by, these other kind of morphological atypicalities rather than the phenotype you care about. And I think that's why, having a common coordinate framework is super crucial.
Definitely you need a common coordinate framework. I'm not saying you.
Or like a 3D common coordinate.
I definitely think that whether you need the exact, like the typical 3D, like that, this vessel is next to this vessel, kind of like in this position, like whether it's rigid. I think like that this is like different. And once we think about it as a rigid structure, we're limited to 3D space. What I'm saying is that also spatially we're in empty space. So it's not a 3D thing, because the distance, for example, in the mouse brain. So when I worked on the mouse, we looked at the somatosensory cortex and have the barrel fields. And no atlas can register the actual position of the barrel field because they change between different mice slightly.
Yes, so that's actually the reason why, we actually wanted that a real 3D reference, so it's easier to match.
No, but you don't. But you need to measure the 3D from that individual mouse in order to spatially map that back. You can't measure from a typical mouse because you're not going to get the right position of the barrels, or to understand like which.
Yes. So I feel like, for example, I don't know, from the experimental perspective, right? So there's always a question when you actually conduct these spatial measurements, it's like which angle to cut, where to cut, right? So there's always an autonomy atlas. But if we have a molecular defined atlas overlay with that, probably we less care about the accuracy of cutting, but it can help restore roughly where you are physically. But I totally agree with you. No two mice are identical. So, but still, it's a lot better comparison across labs, across even the same person who accidentally cut in different variable positions of the mouse.
We had a hand raise from the audience, Zoom audience. So, could you unmute?
No, I mean, I was just listening to a discussion where, essentially there was.
Oh, we can't see.
Oh, sorry, your voice is too small. Do you mind to type your questions if they're functional for typing?
Or is your mic
There's a QA. There's a
Maybe-
QA.
Maybe this is better.
Oh, this is much better. Yeah.
Okay. Yeah, I have no. What's this? Said that maybe it's interesting to have like a big atlas of H&E images and then have those to reconstruct the 3D, like basically have a 3D reconstruction out of them. There have been several methods which are used on specifically like cancer-based, large atlases from TCG and CPTAC, where they have tried to reconstruct the tissue block, based on the entire 2D scan itself. But the problem being that there's no real way to validate it. So what might be actually interesting is that, for example, just to see if we can find certain image biomarkers which can, detect these individual cell types that are usually not seen on these H&E images, right? So maybe there is a way, like in the tissue image analysis community, where we can use these methods, combining them with the spatial gene expressions as well as like possible deconvolutions to figure out like if these cell types actually exist. And if they exist, then is there like a simple biomarker on the H&E tissues itself that could be quite beneficial, right?
Yeah, thanks.
Is that requires like a paired H image and spatial?
Sorry, I couldn't hear you. Can you repeat again? My bad.
Sorry, is that required then to have a paired H image and transcriptomic data to be able to infer the cell type based on the H image?
Well, yeah, but like not necessarily just the H&E image. Even if you have like a fluorescence-based image, that would still be fine, because there are still certain methods which are used in the image analysis community for, just detecting or like segmenting out the nuclei, or for example, just figuring out certain patterns between different stages of cells and stuff. So Visium being quite popular, that there is a paired H&E image, so that helps quite a lot. The only problem being that we still don't know that how the cells look like, right? So we just know that from the genomics aspect. So is there a way to potentially come up with a biomarker which can be used to figure these out on the images itself, and then use that as a method to register the spatial gene expressions with the morphology itself?
Yeah, thanks. Yeah, I think basically there are a lot of discussion about use some high throughput way of imaging to quickly have a whole atlas, and then low throughput, but like high resolution spatial omics to have details. So I think that's kind of like eventually we can get there. Faye, do you have any other comments?
Oh yeah, I wanted to just say something. So we ran this large experiment where we took like 130 sections from a mouse brain for slide-seq and registered it to a paired missile in the mouse CCF, kind of like what's been described. But I feel like you could probably, when we take single cell data and we do batch correction or registration, we're registering it in some latent space.
Yeah.
Probably figure out some tissue latent space where every time you collect some new data, you don't need to register it to CCF. You could register it to like the cell neighbor.
Yeah,
almost certain
exactly.
That once you have that one thing, you could take it'd be an interesting computational experiment, like section the brain in some like really weird, because we section that coronally. Let's section it at like 45 degrees and then ask like, can you place
or flat mount the cortex.
Or flat mount the cortex.
So
That's actually really
I think exactly like we just need to do it once, right? So we know the physical relationship, but then in the end, like the molecular based not the registration, molecular based align or like latent space or something could be even like more powerful, more accurate.
Right. I guess like what I'm saying is that I'm proposing an experiment where we do that and we don't keep track of the orientation, we cut it, or we do keep track of it, but we leave that out and see if we can recapitulate that.
But the foundation is like, it has to be all like organs like a mouse brain where the spatial gene gradients match with some physical feature, but there could be organs it doesn't relevant, like like the physical orientation wasn't correlating with molecular gradients, then we couldn't do that. So that's like also maybe organ dependent.
Yeah, where you have just recurring functional units like in the kidney, in the liver,
Yeah.
in the lung.
But then you don't care about the registration, you only care about the organizing unit.
Yeah, so in the end, it's like a unified
Sorry, I don't know if
molecular
that's true.
coordinates.
I think it's
just-
not quite true. I mean,
okay.
there are clearly diseases that like in the lung manifest
Yeah, only
more
in
the
certain-
upper,
yeah.
lower lobe and that sort of thing. And we're not yet very good at understanding that.
Right, so maybe it's, yeah.
Maybe that's an interesting like goal for the human cell atlas, kind of like that. In fact, like again, just to make it clear, again, I don't also believe some of my statements. Like so, it's quite, if you know me, it makes sense. Anyway, but I'm saying like I do think that you need like to have the reference atlas that is as accurate as possible in 3D. I'm just saying that later.
Later, yes.
And for the mouse, and we only have that for the mouse.
Right.
But we actually, so maybe the human cell atlas, like the next goal should be actually to generate like the next generation of like 3D, spatial, multimodal, H&E representations of all the organs in the human body. Then we can actually find out whether for a specific experiment, we need to actually physically align it, or can we use like the landmarks to what we do.
Right, so I think probably the so-called CCF is not traditionally defined like the image-based CCF. It's also like molecular CCF.
Yeah.
This is what we got in the end. It's like that CCF has additional spatial information than just a single cell-based atlas, but it's not rigid. It's like a mixture of molecular thing plus some spatial information. That spatial information could be at a small scale of basic tissue units, or could be at a larger scale of green slice. So
Can I,
Please
can I
go
ask a
ahead.
question?
Yeah, sure.
So I believe that like the accurate, very accurate 3D position will be the ultimate goal. But I really agree that like we can find the real 3D. Currently it's only 2D. So can I hear your thoughts about when you do cryo sectioning, maybe when you do like 5 micron, one cell maybe be cut like to 3 or 4, 5 layers. How do you have any thoughts like how to integrate like these 5 slides, this one cell? How do you integrate these 1 cells? Forget about the position, like just this 1 cells, how to 5 portion of the 1 cell, like 3 portion of the 1 cell, even 2 portion of the 1 cell. How do you have an idea how to integrate?
I think that sounds difficult, right? So it's like you are having that SEM
Yeah,
action, right? So it's like if you have 1 cell, you cut it into several pieces across several slides. That's hard. So that's why kind of like one of the 3D mapping thing I was thinking is like, okay, one reason we need a 3D is like the cutting is so tedious, right? So if we could cut a thicker tissue slice, that's actually easier for cutting and easier to recover an intact cell. If we cut as thin as possible, we do have a lot of cells. They just like fragmented across the slides. I guess it may be hard to actually reconstruct an intact cell when they spanning many slides. So,
yeah, but
I think
no
like
matter how thin,
yeah,
and no matter how thin or how thick, there's always a lot of cells be cut. Like a single cell, a lot of like cell missed or die.
Right,
This
I
like a
think-
lot of cell be cut. I think the ultimate goal, eventually, I think we'll figure out how to integrate this 2 part or 3 part of the cell, right? But you have any ideas? Just
that's like right. So if we could cut like 10 layers of cells all at once, we only have small fraction of the cells fragmented. So that's another value of 3D, if it's possible. Okay. So maybe we should move on. So I guess for the like session 2, since our time is limited, but I think also we divided into 4 questions, but it's actually just one. It's like we go from molecular atlas to function and dysfunction. And whether the function is defined by, for example, functional imaging, or it could be like electrophysiology measurements, it could be even some mechanical properties, cell morphology information. And then we we could maybe use that to first uncover some basic mechanisms, and second, disease. And regarding disease, because like even with a wild type like regular human, so we have a lot of variability. But then we study disease, right? So we have to draw a conclusion, this is disease, this is control. And then typically, given the large variability, how do we decide how many replicates we need to address the variability and to make a meaningful comparison between disease and control? So that's kind of like additional challenge besides a version 1 atlas. Yes. Any functional perspective and any disease perspective about spatial technology?
So I'm not a spatial person, but the form to function or genotype to phenotype question is sort of like one of the oldest, math that we've been trying to build over the years. So I guess my question would be like, I wonder whether 3D would give us a little better linkage. I mean, this was already sort of touched upon. If you were able to take the brain and flatten it out, identify certain regions, or she said the lungs have a certain region that always has a disease, then maybe we can have a targeted cell population for therapies. Maybe space can give us a resolution we never thought about. But I'm not sure how space helps us link this form and function.
I guess that is a good point. If we want to know the function of either like this thing, that need to be 3D, I guess. The 2D may not be like truly show the function of that cells or cell, multiple cells.
Or, for example, we need to define what do we mean by function, right? So it's like, I think probably when we mention about function, it means like at a single cell level, whether the cell is functioning as expected and what's their major functional result. It could be, for example, cardiomyocytes, they're beating, right? So neurons, they're firing. So like different cells, they have their defined single cell function. That's like a level 1 function. And level 2 function is like the whole cell population work together to provide the tissue function. And so I think probably there are a lot of variety of what do we mean by function, and we can try to like matching individual functional definition with a particular molecular feature, and eventually overlay them.
I'm Diane Saunders. I'm one of the Pancreas By Network coordinators. So I mean, not to like state the obvious here, but really I think the ground truth is if you can do multiple modalities on the same sample, because humans are so variable. So for example, in our organ, we can isolate the functional tissue unit, which is an islet, and that's composed of a bunch of different cells, and we can further disperse those cells and do patch-seq experiments on them, and then kind of back that up and correlate gene expression, other kind of high throughput assays in some type of modular format where you're collapsing essentially the variability down into discrete chunks that can then be mathematically distributed and probabilities can be calculated. So I think like having something like that as the ground truth before you can then build models to apply that to single modality experiments is going to be really important, and that'll obviously depend. The workflow will depend on the organ.
Yeah.
I'd really like your comments about function. I think I'd like to make a further distinction between the way you define function and mere association between genotype and phenotype.
Yes.
So usually in function, it's like the classic things like you knock something out and then something doesn't work and you say that thing is causing that thing. So we have this intuitive idea of function as like a counterfactual, right? That's how we think about it. Or you upregulate a gene and some phenotype appears, that's function. So I think from like perturbation in, say, the genotype, I'm speaking very broadly, to a change in a phenotype, that implies function. But I think an association between the genotype and the phenotype, from there it's hard to infer function backwards. So I think the logic doesn't go from genotype, phenotype to function, but it does from function to genotype and phenotype. So I think in the spatial technologies, because we are not perturbing the system directly, unless we are like comparing disease cases or something like that, what we're actually doing, in my view, is associating, whatever modality we measure with a phenotype of interest. And I think the next step would be that, okay, let's try to do an actual experiment, like the old biology, to figure out function. Yeah, so I think, there's a sharp distinction in my head between function as classically understood and an association between phenotype and phenotype.
Yeah, I think that
limits the space of
right. So
possible
I think that's definitely there's like association, correlation versus the final causality and perturbation. So I think they're like two steps. But I kind of like want to take in the patch seq as an example. I think that's kind of like a perfect example for single cell level function, right? So how the ion channels and other genes correlate with a particular physiology feature. But the beauty of the patch seq is like, for example, if we do gene perturbation, it's one gene at a time, and that gene knockout could have collateral damage that not actually that gene, right? So they are in the development. But the nice thing about patch seq is like you can actually infer largely like which ion channel is responsible for which type of electrophysiology feature. So you kind of like have a global discovery of gene programs and genes to particular functional features. But then you can come back to perturbation and validation. That could be much high throughput and easier. You just need one single experiment instead of doing so many repeated knockout. I think that kind of like cross modality gene to function correlation will be a first hypothesis generation type of thing, and then we can go back to the perturbation side.
I guess on the case control side, I just think about what we do for single cell, which is like, well, you register the data in some common space, and then you look at maybe differential compositions of cell types or states and differential gene expression between case and controls. And so really to be able to do this correlative analysis of genotype to phenotype, right? Where genotype is like case, there's some genotype now, it's like case versus control. We need some like way to register cases and controls in some latent space to compare them. And then we'd like to compare like changes in composition, interaction, gene expression changes. But it'd be nice if we had this like representation of the tissue we talked about, where then you could.
I mean, the pathologists, I don't know whether they're pathologists in the audience, but they must already have a huge bank of pathology images that are annotated, that are being used, being analyzed with machine learning algorithms to,
Well, yeah, that's true. But most of their annotation is some human intuition about, what's going on with the.
It's usually semi-quantitative.
Yeah. Or some quantitative, empirical features about the pathology, right? But it'd be nice if we could. The point of the atlas is like to collect a healthy reference, in the same way that in single cell we can, take a healthy reference and just align the single cell data collected from disease to it to see the new states. Like it'd be nice if we could align a disease tissue to it and see like the new compositions or the new interactions or like the new neighborhoods.
I think what you mean, like in terms of like patch seq is really like that, kind of like the ultimate experiment, but it's really hard to do.
Yeah, very low throughput.
It's extremely difficult, like to do, right? And you cannot do it, like maybe you can do it. But it's kind of like I always feel like the functional part is like the part that we don't even dare to dream to get. Like in the sense that, for example, actually there are methods in neuroscience like that you can record whether the cell has done like a calcium event, like or a memory engram formation of these things, and maybe translating these, because calcium does everything for every cell. So maybe kind of adding that, like the calcium like signature of a specific can tell us something about the memory or the experience that the cell has.
So I think that's kind of like one thing the spatial technology can offer, right? So because it's already in the format of imaging, so that is much easier to register with other imaging. Calcium is definitely some universal messenger, so that's nice. And but it's a little bit hard to do calcium in human. So I think it's good for a lot of like model organisms, zebrafish, mouse, but it's a little bit hard to do in human. So that we have a collaboration. We actually like electrode measurements. We actually try to measure in like electrophysiology in CT. We don't have image, but we know where the device is, so we can register like spatial. Like,
do you need to find the same cells, or can you infer the activity from?
Yeah, because the electrode is cut together with the tissue, so we know exactly which cell they're recording. So hopefully that could have some medical application. But I think that the general theme here is like different organs need different functional readout. Some cells are just self-sufficient, just based on transcriptomic feature resolution. But by the spatial omics, maybe we can already know what's the function of the cell, like if they are like immune cells and many other things. But there could be some other organs, as I mentioned. Some organs rely on circuits of cells. Right? So we need a lot of cells function together. Then that's the place we need some like organ-wide functional characterization or circuits level functional characterization. So.
Yeah, I mean, I just want to make, again, probably an obvious comment that like all of these questions are contingent on like what do you consider normal, and the fact that like normal is a large group and or it could be a continuum, or it can be a continuum across multiple traits. And so I think, defining and so there are, consortia, I think including HCA, that are trying to do this. HubMap is another one, but kind of you have to build your power based on just defining the kind of confines of normal. So I think that that's a really important aspect of a lot of this.
Yeah, just to add to the set of things that are complex or that sort of add problems to what we're trying to do. I think there's also the issue of mapping from the biological space to the phenotype space, and it's a many-to-one mapping. So in one of our lab meetings a couple of weeks ago, somebody presented a paper on like Alzheimer's disease, where something goes wrong with the cerebrospinal fluid and the people have Alzheimer's disease. And then someone was saying, hey, but Alzheimer's is like when the tau proteins aggregate or whatever. So those two things seem totally different in the biological space, but they say it causes Alzheimer's in the people, which we think is the same disease, but it may not be, or it might be the same disease being caused by two different things in the biological space. So in these associations, there's this other problem of mapping. So it's one thing to be like two things are associated, but in the same phenotype is probably associated with a bunch of other things that can go wrong in the biological space. So that's like, oh, here's another thing that we have like not that much like knowledge about.
Yeah, because disease are defined by function, or dysfunction. So which state may not be, with accurate molecular correspondence. It's just like, but like one thing we didn't discuss here is like how spatial technology can also help with, drug discovery, organoids, and this type of thing. Regarding your comments on Alzheimer's disease, yes, there are so many hypotheses, but eventually, when we can cure the disease, that's kind of like the measure of whether all those like was share the mechanism of those disease. But I guess no matter what, like the spatial omics definitely offer more information than people. Like for example, they can generate different knockout models for Alzheimer's disease or transgenic models for Alzheimer's disease. But previously they just have this like phenotype readout, but now they have spatial omics readout. Maybe they found something in common, and which could generate new therapeutic hypotheses. So.
I guess one of the things, it's not only for the spatial transcriptomics, but how to align with the previous knowledge, like previous experimental results, which done by many animal studies or something like that, and or maybe histological annotation by pathologists. So then we could maybe automatically more discover
yeah, so they're definitely like for pathology has been used for so long, and although it's not that molecularly accurate, but there are a lot of values of that. So I think correlating spatial new spatial data with the old spatial like a phenotype or other experiments-based characterization is actually very important. So we put ourselves in the history of all this medical research. So.
can I ask, can I go back to functional unit a little bit? You have 5 minutes more. I think this is very important to define the functional unit from the spatial data. And there was a discussion this morning that how can we decide the size of the neighbors, 1 cells or multiple cells, or so. I think it depends on the organs or depends on the condition how the size relates to function or phenotype. So is there any like idea or thoughts? How can we like not by feeling but by some sort of
Yeah, some spatial omics definition of functional units.
I think you need to measure all of them. Like in all your experiments, you need to play like with you need to kind of like try to measure the entire like length of resolution. Like the problem again is again like when you get to very large neighborhoods, then they're not really well defined. So and then actually you need the CCM. Like when you're going because in the brain, just taking like a spot in the brain and then extending it. Like once you reach 50 microns, then you're taking multiple layers in different proportions. And it completely doesn't make any sense. But if you take like a cortical column, along a cortical column, then it makes a lot of sense in that way. So I would say people, I would encourage people to try to go from the minimal like single cell cell-to-cell interactions to as much as they can, as long as they have like a good reason for extending that axis in that direction. Just don't take like a circle, and just like the random linear neighbors. Once you go to the macromorphological features.
Thanks. Yeah, really interesting discussions. I mean, I think my take on this would be, and I agree with you, but doesn't this always come back to what your research question is every time? Because these technologies are so powerful now. We're now into a realm of how much can you sequence and at what resolution. So, the way we approach it in Liver, we are deliberately simple about it is, what's the question? And then that guides, for example, what you regard as the functional unit. So, and we're all feeling our way with these amazing technologies, but I think, sometimes it's useful to just drag yourself back to that every time. Just comment.
Yeah, the last week, one of the big paper on the human cellular breast paper published, used every technology on the earth. That's how their last sentence in the discussion. Although our atlas has identified a large number of cell states and validated them in situ, future studies will be needed to validate their functional roles. So we are still back to square one with all the technologies. So I think this is a question maybe next generation will ask. I'm not sure.
Yeah.
I'm not trying to be pessimistic here, but there's got to be that.
So I guess I'm really looking forward to the computational discussion of the spatial technology, because that's like if we have the resource, unlimited resource, we just mapping all of them with all technologies and computationally integrate them.
Yeah, well, I mean, my question was more about like, what is the question we could answer? I mean, for me, if you just, what is the maximum number of cells that could sort of work together? Now, I know nothing in terms of how cells act, but we know a lot about, let's say, social systems, and there's a lot of theories about humans, how many humans can work together that work functionally without needing extra level. Maybe cells have some limit. There's some studies that believe 150 humans function without too many extra levels of policies, but maybe cells have the same things. Yeah. Maybe there's the minimum functional unit could be 100 cells or 50 cells, and then maybe we can identify that. And then that could tell you what kind of organ you might be.
I mean, in a fun way, what's your readout on that? How do you assess the quantity? What, and I know this gets to the heart of what you're saying, but how do you define what a functional unit might be? What are your parameters for trying to understand that? And I think that's really cool, because that's, a very unbiased approach. And obviously our research question is a very focused approach. But in a cool way, I think, everybody's looking at this from many angles.
So I think that's really a great foundation question, right? So it's like, what's the actually smallest like functional cell community that they were able to communicate with each other and coordinate with each other. I think that may be true for many like things like think about skin and like they are like a repeating units. The thing about the brain, I feel is the whole brain, right? So they connect with everything together. So, but I think all this spatial omics analysis will help us to actually learn some principles, like what's the basic or different organization principles of the cellular community that they can play a basal tissue functions.
Yeah, I think you can kind of even start with like cell to cell. So I would divide the problem as cells that move and cells that stay where
Yes,
they are.
yeah, that's also very important.
So it's like immune cells are moving around. So it's like you get a snapshot, but neurons don't move around. So that's like pretty set in stone. So that's okay. So let's just stick with the neurons because it's easier. But then the problem is they go, they spread out really far. So let's just look at the ones that don't have their axons going all the way, and then there's some more like localized. I think by kind of circumscribing.
Yeah, I feel that's kind of like many people try to utilize some like ecology analysis in spatial omics. So which means like we have plants and animals. Plants like plant cells don't move, animal cells they move around. So I think eventually it's really like really fun like cellular ecology problem we are talking about.
Maybe the last.
Yes, the last.
My name is Javier. I'm from the Schwartz lab. I just wanted to comment about the finding this minimal functional unit, and just like even in some experiments, you can see like, oh, we see some type of interaction or some type of communication at the level of, let's say, 5 cells. And then you might think like 6, 7, 8, it appears to be the same type of communication, but then you reach 10, and then the communication changes, the patterns, the circuits. So again, it becomes really complicated, right? Like even defining that functional unit, right? Because the unit changes.
Yeah, so I think probably even the cell don't move, right? You can send chemicals everywhere. So I think they're like a different spatial scale we are talking about. So yeah, I think it's again like some computational questions like multi-scale models for like the spatial omics because they have different spatial scale of communication. Okay, great. Thanks for the great discussion.